What is black and white, black and white, black and white, and green? What is black and white, black and white, black and white, and green? A skunk eating a pickle, of course. 3 skunks eating a pickle. 3 skunks. Black and white, black and white, black and white. So you have to remember, some years ago, my girls actually wrote some of these to try to help me out. So that one sounds like one that they constructed themselves. What do you get when you cross a caterpillar and a parrot? Caterpillar and a parrot. A walkie-talkie. Where do crayons go on vacation? Where do crayons go on vacation? The Wax Museum. Oh boy, you guys have some studying to do, because what's happening a week from today? Exam number 1. A week from today. So I know a lot of you have been staying up on the material, which is awesome. Some of you haven't, which now is the time to kind of lean in this week. It's Monday, so you have the opportunity to really kind of make the most out of this week. So a couple of things this week. Number 1, we've got 4 review sessions. You ready? The first one is Wednesday, 11 to 12, Science Annex 122. And that's Eliana's. Eliana also hosting one on Thursday, 5 to 6 p.m., Science Annex 122. And then Amara on Thursday, 6 to 7 p.m. in Weta 242. And then Sunday, you get both of them in the afternoon, 3 to 4 p.m. This room, Bio 265, on Sunday. That's the day before the exam. And that's at 3 o'clock in the afternoon. That one will be on Zoom. If you can't attend on Sunday, but you can Zoom in. Okay, and we'll try to record it as well on Sunday afternoon in case, everything fails, right? You can't make any one of those 4. So here's how this works. And you're going to want to stay the whole time. We're going to have a participation activity today in class. You're going to also have the opportunity for 5 participation points by going to one of those 4 review sessions. You just have to go to one of 4. Okay? You don't have to go to all of them. But basically, we don't take attendance, but we're providing what we call some easy opportunity points that move you through the studying process. Make sense? So 5 points for today, just stick till the end. 5 points for participating in 1 out of the 4 sessions before exam 1. Question?
Sunday, you could either come in classroom or on Zoom.
That's right. Zoom. You could Zoom in on Sunday as well. And we'll post that, and that will count as participating. Okay? Any questions about that schedule or that rhythm? Okay, cool. If you also remember from the first day of class, we talked about bonus opportunities. Those are separate than participation. Participation, like today's participation, is 5 out of 5. And going to one of the review sessions is 5 out of 5. So there's 10 total participation points for Unit 1. There's also a bonus opportunity which hasn't shown up yet. And there's only 1 more class period other than today before the first exam. So we'll let you guys use some deductive reasoning on what day that probably is going to be. Okay? Any questions on how the bonus opportunities work? I'll have to explain the grading. I won't do that today. We'll explain it more on Wednesday. But Canvas is a little weird because you can't make a bonus opportunity worth 0 on Canvas. Everything is worth points. The old system, you could do that. So we have to do sort of some finagling in the grade book. But you'll see your bonus opportunities. You'll see your exam 1 score, and then you'll see the 2 scores combined. Right? Bonus opportunity 1 plus exam 1 is your total exam score for Unit 1. Does that make sense? I'll explain it again more on Wednesday. One adjustment to the schedule. So September 30th, which is 2 weeks after exam one, that's a Monday, so it's 3 weeks from today, we will be doing an online lecture. I actually have to be gone. That one just came up. Anybody have any more troubles with accessing the GRL textbook? We get all those things cleared up? Anyone? Everything's going smooth? So what's happening this Friday? Quiz what? 4. You don't have to wait till Friday, but it expires at 11:59 p.m. Okay, you all feeling pretty comfortable with the material to date? I would look at your notes. I was walking down the aisle. I got a new microphone, so hopefully we won't have any more AV issues. And actually, it's a new iPad, so hopefully it'll be smoother, maybe a little better quality even. The last camera was fine, but they always make newer iPads with better cameras. I'm walking back down. I'm seeing tablets and iPads, people taking notes right on the lecture slides. That's probably my favorite way to do it. Maybe my second favorite way is you don't have a tablet or an iPad. I know they're expensive. You print out maybe at the library. Yeah, it wastes paper, but don't consider it a waste. You could do double-sided. You can do 2 slides per page, and you take written notes on the printed pages. Another strategy is just have a notepad and you take notes on the slides and you explain kind of what the slide is, and then you reconcile that later with the downloaded images. Because what you really should be studying for exam 1 isn't the quizzes, it's not the reading, it's actually the lectures and the lecture slides and your notes. Let me repeat that. What you should be studying for next week's exam are the lectures, the lecture slides, and the lecture notes that you take. Make sense? So it's why I go through all the trouble to try to record these lectures. Every now and then we have a glitch and I have to use a last semester recording or an older recording, but they're basically the same. They're just a different lecture hall, or maybe I have a tie on that day, and I am about 6 months younger, so there's that. But maybe I'm getting better. I don't know. So the lecture slides and the lectures that are on YouTube are my favorite modality to help encourage you to prepare for the exam. We will have some reviews, guides that we'll post on Canvas. We'll open those up. Here's the danger. They are not to replace. They are to reinforce the lectures. So if you just go to the study guides and you study just the study guides, there may be something in lecture that didn't make it in the study guide. The study guide is a good tool. As you're studying, you go through it and say, is there anything here that I'm totally unaware of? Did I miss a topic that he covered that's on a study guide that I actually either didn't go to that lecture, I don't have great notes? That's the way that I would use that, is maybe a way to quiz yourself or quiz your study partner. Okay? Is it okay to go through the study guide and try to answer it? Absolutely. But again, it's not to replace the lectures, it's to reinforce. Make sense? Okay, so to date, we've been covering basically review material. And we're going to be in lockstep this week with lab. So this week is lab 1. You're in organization of the body, which we've already talked about, and you're in microscopy and tissues this week in lab. That's correct, right? I've got the right updated schedule? That's what we're doing this week. We have 2 lectures on histology and tissues. And this is a huge adjustment from where we've been, because we're not doing a bunch of review. We're not talking about the foundational building blocks anymore. We're actually utilizing them, and we're going to be talking about content. And on this first slide, you can appreciate some of the histology that you're going to be seeing as you go to lab this week. Upper left, areolar connective tissue. In the upper left, we'll come back to that picture, but there's a couple of different types of proteins there. And the connective tissue is very matrix rich. and cellular pore. We'll talk about that again. And then on the lower right, you have a picture of bone, which is also a connective tissue, a type of connective tissue. We won't get into bone until Wednesday, but we're going to set up epithelium and connective tissue today. And the way that we're going to look at this is using histological insight. So what is histology? Histology, is simply put, is microanatomy. So you remember in one of the early lectures I talked about in the 1600s, the invention of the light microscope, and we looked at cork. And now all of a sudden we started asking all these new questions because we saw the architecture of cork. And when you see the architecture of cork under a microscope, and there's all these little boxes, you can't really appreciate that with the naked eye. And when you look histologically at microanatomy, and that's really the definition of histology, is microanatomy, is you get some insight as to the architecture and the structure of the tissue. And remember, this class and lab, we're big on structure leads to specific types of function. And the structure and the architecture is built and designed a certain way so to elucidate a very specific type of function or physiology, a purpose. You know, we talked about us being consisting of small cells and not one giant big cell. We're specialized. One of you actually said specialization of cellular architecture because we have different functions. That's kind of the beauty that makes up humans. Tissue. Tissue is defined as a collection of cells performing the same function. So we'll look at different types of tissue, like epithelial tissue, connective tissue. And then an organ is a collection of two or more of these tissues. Like, so for example, take skin. You'll have a portion of connective tissue, which is the underlayment of skin or the deep tissue. And as you move superficial, you move to epithelial tissue. So two or more different types of tissue make up an organ. So the skin is the largest organ in the body. Upper right is a picture of pseudostratified ciliated columnar epithelium. That's a mouthful. You'll see it again in lab. Pseudostratified ciliated columnar epithelium. So pseudostratified, because pseudo means what? What's the word pseudo mean? False or fake. So if it's pseudo, it's not really stratified tissue. It's just that those nuclei, instead of all being lined up, that would be simple tissue. Stratified is where you've got multiple layers, so you see the stacking of nuclei. But here, they're actually just narrow cells and then wider cells. And so the nucleus actually sits in different levels of the cell, but it's still one layer. So it looks stratified, but it's not really stratified, so we call it pseudostratified. Ciliated, that's little hairs that are on the surface. You can appreciate those little hairs where the arrow is pointing. Pseudostratified ciliated, columnar is the shape. So we're talking about squamous, which is flat. We'll talk about columnar, which is like a column. We'll talk about cuboidal, which is a what? Cube, or more of a square, okay? If you lean into this, it's actually pretty easy. Like the name is descriptive. Scientists and engineers are not all that creative, right? We call it what we think it looks like, or maybe what it does. The lower right, any guesses as to what this is? If you guess it, I'm impressed. But if you looked at the readings like I asked you to, perused them, you might have seen this. What kind of tissue do you think it is? Let's start there. Purple tissue, Keller. I mean, it's not bad to look at the colors. But I will warn you, every now and then we'll get colorblind folks in the class, and actually they do the best on histology because the color can actually distract you. If you focus on the architecture and the shape, you actually do better. So don't think of it as purple tissue, but oftentimes neuronal tissue is stained this color because the hematoxylin is taken up nicely by the neuron. That's a neuron. Okay, 4 different types of tissues between today and Wednesday that we're going to get into. And as I explain this, I'm just going to check my recording to make sure we're on. I don't have another snafu like last time. Okay, we look good. So epithelia, nervous, muscle, and connective. The 4 different types of tissue. Now, what's this column on the embryo? Well, it's where they come from. So in the embryo, you have a couple of different layers. And the epithelial layer, or what we would refer to as more of the ectoderm layer, gives rise to epithelia and nervous tissue. The mesenchyme is the middle layer. And the mesenchyme gives rise to muscle and connective tissue. So they just have slightly different origins from an embryonic development standpoint. The book does a nice job of explaining this. It goes into a little bit of stem cells as well, and what stem cells are and how they behave in the different categories. But again, this is where I'm trying to get you to focus. Focus for the exam on the lecture material. So, well, why do we even bother with the textbook then, Keller? Well, we give you both a broad exposure to the material through the textbook and the quizzes, and then I kind of focus and I go deeper into the physiological aspects of what we cover in lecture. And every professor has their preferences, right? They emphasize different things. So I think one of the best ways to guide you is for the exam, study the lectures. And then I'll write my test questions from the lectures. So 4 different types of tissue. Nice potential test question there. Right? Some examples like epithelial tissue lines cavities or it covers the body, like epithelium. The topmost layer of the skin is the epithelium. Nervous tissue like the brain and spinal cord. Muscles, they're for movement. They facilitate locomotion. Somewhat self-explanatory. Connective tissue, some example like bones. I said that bone is a connective tissue. Well, tendons and ligaments are also types of connective tissue. And we'll dissect those down physiologically in lecture as we move forward. So a couple of things that I think will be helpful in lab for you to consider is that the way that histology happens is you take a biopsy, you take a sample of tissue, you take all the water out. You take all the water out by dehydrating it, going through a series of alcohol, ethanol rinses. So you'll start with a 70%, which is 70% ethanol and 30% water. Then you move up to 80, then 90, then 95, then 100 twice. You usually do that for 30 minutes per cycle. And when you get to 100% ethanol, now all the water is out of the tissue. And then you infiltrate it with wax. You perfuse it with wax. You vacuum infiltrate every aspect of the tissue with wax to hold it together. Then you slice it really, really thin, usually about 6 microns, less than 10 microns. You put it on a glass slide. And now you can actually look at things. You have to stain it with different stains, and that's where we get these colors. So the cross sections can be deceiving. Very deceiving, right? So if you look at panel A and you put this little, I think it looks like a noodle, okay, like an elbow noodle. You put this in a wax mold, and depending upon where you slice, like if you slice way up top here on this red first dotted line, or orange, I guess it is, you're going to see 2 circles. Make sense? Because you're cutting cross-sectionally, and you cut through this circle, and then you cut through this circle. You cut through this green one, the second dotted line or the middle dotted line, you actually kind of get this funky structure that's connecting or bridging the 2 circles. You cut through the third or the bottom one, you just get the end. So in histology, it really depends on the orientation of what you're seeing because you're getting a snapshot as you've cut through the material. So a lot of times we say, let's do serial sections. What's serial sections? That would be cutting all the way through the noodle and using all three of these, which are in series, and you look at them collectively and you make your decisions. Make sense? So having quality histology is almost as much of an art form as it is in a science, right? And you have to have an appreciation for 3D visualization. So another exercise over here, if you cut right through this, you can kind of appreciate how you're going to get very different shapes depending upon the orientation of the slice. Down at the bottom of this slide, we've got some histology examples. And I want you to test yourself. What do you think is on the far left, your far left, histologically? Any guesses what kind of tissue that is? Maybe you haven't had lab yet. That's okay. Any guesses? What's that? It is not bone, but it's very close to bone, in close proximity. It's not marrow. It's a good guess. It usually is on the top of long bones before they articulate to another. Cartilage. So it's cartilage. It's actually hyaline cartilage. What about the next one over, this pinkish looking one? What's that? It's a connective tissue. It's also cartilage. It's also hyaline cartilage. Okay. The pink one, the lighter pink one, what is that one? It's cartilage. It's hyaline cartilage. Wow, you guys are catching on. How about this far right one? Kind of the black and white looking one. It's cartilage. It is hyaline cartilage. So remember how I said, like, don't be deceived by the color. All hyaline cartilage stained with different stains. It's like, wow, I am really confused, Keller. Well, what color is tissue? When I take it out of the body, I dehydrate it, I infiltrate it with wax, I slice it really thin, I put it on a glass microscope slide, and I hold it up. What color is it? Any guesses? Pink? What's that? Clear? Pink or clear? Any other guesses? Huh? White? How about some ethnically diverse answers? What do you think? Like, ooh, he went there. Huh? Whatever color's behind it, whatever the color of the skin of the individual. Is that what you guys think? Guess what? It's a pretty diverse group in here. All of our tissue that's sectioned would be essentially the same color. Doesn't matter your ethnic background. Relatively clear. Because tissue is basically colorless. You have to add the stain to be able to see something. So in this sample, this is skin. This is skin that's very lightly stained. It's got a hematoxylin, a light hematoxylin that helps light the nuclei, which are the round structures that pop it a little bit subtly purple. And then if you can appreciate some of the brown staining, that's actually what gives skin its pigment. We'll talk more about that later. That's melanin. So depending upon your ethnic background, there might be a little bit more melanin or there might be less melanin, but everything else is relatively clear. Then what we do is we stain it aggressively and it looks like this. So we can actually see structures. But I always love kind of using that example of at the basic level, our skin is all the same. The shades of, or the tone of color that's in our skin does come from melanocytes producing melanin, which gives it a pigment. But histologically, it's very, very subtle. And we have to do dark stains to actually highlight, no matter what the ethnic background is of the patient, highlight certain features to come out. These are both skin preparations. Okay? All right, epithelial tissue. That's kind of the introduction to tissues to try to get you kind of oriented for lab as well. On, you go pick up a microscope slide. I wanted you to know how it was made and what's all behind it. There's a lot of technique behind it. So epithelial tissue. Epithelial tissue is our first category. What does it do? Epithelial tissue forms these 4 different functions, or provides these 4 different functions: protection or a barrier of some level, absorption, secretion, or filtration. Not all of them do all the same things, but epithelial tissue as a category has these characteristics associated with it. I think protection or a barrier, it's pretty easy to think about, like outer facing tissues or surfaces like the skin. That's a barrier to the outside. It actually prevents bacteria, viruses, and fungus from entering into your system. That's why if you have a cut or a breach of the skin and it stays open chronically, we call it a chronic wound in your patient population, that's a problem because it can become infected. Absorption, also really important in the small intestine. You'll learn about the small intestine next semester in 202, but we'll talk a little bit about these tissues, and you'll have a little bit of review in 202 about tissues before you launch into it. But this is, again, the background of the 201, 202 sequence, or combo of classes. So in the small intestine, you need to absorb nutrition from your digestive tract and bring it across the basement membrane, absorb it into the bloodstream so that you can actually utilize that nutrition that you've consumed. So an epithelium that allows for absorption is really important. Secretion, like in the kidneys and different kind of gland organs. You actually have to secrete products into the small intestine, enzymes to help break down food components. Break down proteins, you have proteases. Break down fats, you have lipases. Break down sugars, you have other aces that break down, like sucrase, right? Breaks down sucrose. Filtration, also in the kidney. You need to filter the blood to eliminate waste products. If you have patients that have poor kidney function, as measured in what we refer to as glomerular filtration rate, or GFR, you'll learn more about that next semester because that's a system that you cover in 202. But that can be at toxic levels, and patients have to go in for dialysis 2 times a week, 3 times a week. They pull the blood out of the body. They run it through a machine, which is basically filtering it, like your air filters that you see in your house or your car. Then they put it right back in to the patient. They sit there in the chair for a few hours. That's the function that your kidney does. You have 2 of them that just follow you around, right? Pretty convenient. So now you can kind of appreciate, we've got simple squamous, simple cuboidal, simple columnar. Simple meaning 1 layer. You'll reinforce that in lab. Stratifying meaning multiple layers. But again, this cartoon diagram, I think, kind of helps to illustrate where they're not multiple layers. It's 1 layer because they're all touching the same basement membrane, but they're shorter or wider, skinnier, right, oblong. And so they kind of have this weird appearance instead of being nice and organized like stratified squamous or stratified cuboidal. Some other characteristics of epithelial tissue. Well, it's cell rich. It's high in cell content. And it's relatively low or poor in matrix composition. So cellular rich and matrix poor. It will make more sense when we look at connective tissue, which is the opposite. Connective tissue is cell poor and matrix rich. Okay, but here, lots of cells. Think about skin, the architecture of skin, densely packed cells. You've got what we call tight junctions and adhering junctions that hold these cells together. In the case of the integument or the skin, you want neighboring skin cells to have tight connections to each other because you don't want something squeezing through. In fact, you actually have specific cells in the skin that look out for foreign invaders. These are referred to as dendritic cells, and we'll talk about them again in the skin system. But they basically phagocytose, or they eat bacteria that crosses through the skin barrier before it reaches your bloodstream. They're like sentinel cells or soldiers that are on the lookout for invaders. So these adhering junctions were referred to as desmosomes. That's the scientific term for an adherent junction. A lot of epithelial tissue is so dense in cells that there's very little room for blood vessels. And so the blood vessels are beneath it in the connective tissue that's underneath it, like in the skin. And through diffusion, an area of high concentration to an area of low concentration, oxygen moves from the blood vessels to the skin. And it moves at about a distance of 250 microns in tissue. That's the extent of how far oxygen will diffuse. Does diffusion require energy? No, it does not. Does osmosis require energy? No. Give me an example of a transport that requires energy. What's that? Active transport. Give me an example of an active transporter that you know about and love and have studied. I see some heads shaking like, not funny. I could make that a flashcard. No, it's not a good joke. Okay. What's that? Any takers? An active transport that moves 3 somethings out and 2 somethings in. Keep going. Sodium, potassium, pump, or ATPase. Either one of those terminologies would work. Okay, great. Characteristics of epithelial tissue continued. Well, they're sided. They have a basal level and they have an apical level. They're polarized. That's all that word means, is polarized. Like, the election's coming up. So you hear language like, well, these 2 individuals are polarized on this topic. That means they're on different sides. They don't agree. So polarized is just a word that means sided. Basal side is the basement side. That's typically supported by connective tissue and has the basement membrane. The apical side faces the lumen or faces out, and that's the site of secretion, like where the cilia sit or the microvilli. If it's in the case of secretion, we've got 2 different types of secretion in the body. We've got what we refer to as exocrine. Exocrine, like exit, E-X-O. So I think of exit, that means you're releasing it outside of the body or into a lumen. That lumen could be continuous with the outside of the body, like the small intestine. Now think about this. This kind of blows some students' minds. You consume food. You had a salad for lunch. You chew it, which is called mastication. You'll learn about that next semester. You start digestion, which is chemical breakdown. Mastication is the physical chewing, a physical breakdown of food that starts in the buccal cavity or the oral cavity. And then you release enzymes to start digesting the food, and then it moves through the esophagus to the stomach, through the alimentary canal, GI tract, eventually out the anus. The whole time it's inside that lumen, it's part of the outside environment. It's only until you absorb it across the basement membrane that it becomes part of you, and it's on the inside. Does that make sense? So now you think about some foods that are irritable. You eat something that doesn't agree with you. What does your body do? Well, let's get that through as quick as possible, right? Makes sense. Because it's potentially toxic. I don't want to absorb it, so let's just flush it through. And that's why GI motility is high in patients with diarrhea. So exocrine, releasing into a lumen or outside of the body via some sort of cell, like example would be a goblet cell, where it releases mucus. Or endocrine, which is it releases it into a duct or secretes it into the bloodstream. Endo meaning inside. Endo is a prefix that means in. So we've got 2 different histologies here. I'm just trying to give you a few examples to try to reinforce what you're going to be seeing in lab. Upper right, you've seen this before. What do you think that is? Upper right. Pseudostratified, ciliated, shape, cuboidal, simple, or columnar epithelium. It's the long one. You get that one, you're going to have no trouble with any of the others, I promise. Okay? How about this lower right? I was like, what am I looking at? It's like green with brown dots. Like, remember, try to ignore the color. Just look at the shape. These are nuclei. The brown is staining the nuclei. Do they look like cubes? Do they look like columns? Do they look flat? Yes, they look flat. This is a lumen. What do you think that is? Is it multiple layers on top? It is? Is it multiple layers, or are they all in the same plane? They're all in the same plane, so it's simple, not stratified. Simple, and it's flat. What's the word for flat? Squamous or squamous, yeah. So on the exam, if I'm going to ask histology questions, they will be pictures that you've seen in lecture. Okay? And they'll be on the screen, right? The way that'll look is probably 5 to 7 pictures that will rotate. The last part of the exam will be microscopy or gross images or image-based questions that you'll see from your seat. But if you're here in lecture, you're reviewing your lecture notes, you will recognize these pictures, or you should recognize these pictures, okay? Fair? Okay, characteristics of epithelial tissue. Rapid cell division. So the skin actually goes through an amazing turnover. An amazing turnover. In about 2 months' time, 2 to 3 months' time, you'll replace the entire integument. It starts from the basal level, it moves up, and it sloughs off. So there's rapid regeneration. And on this slide, here's the biopsy that we're taking. And so the outside is on this side of the screen, and there's a dead squamous zone here, and then there's a living epithelium, and then this is the underlying connective tissue. And so that's under constant stress, these tissues, and so they need to turn over. Epithelial tissue needs to turn over on a regular basis. And we'll get into more of that turnover in weeks to come. There's other kind of categories or characteristics of epithelial tissue that aren't quite as convenient as what we've been talking about with simple squamous, simple columnar, simple cuboidal. We've got pseudostratified, which is always ciliated. I introduced you to that one. You guys seem to be an expert on it already, which will be great for lab this week. That's the top picture, pseudostratified, columnar. This one's actually from the trachea, this particular preparation. So what do you think those little cilia do in the trachea? What do you think they do? What do you think they do? They bring up mucus, right? You're like, wow, what's wrong with him? I was trying to demonstrate. So when you cough, a little bit of mucus comes back up this respiratory escalator, keeping that debris out of your lungs. It's exaggerated because the goblet cells make extra mucus when you're sick, and that's when you get really phlegmy and you got like loogies all the time. But even right now, if you just did a little cough, you won't notice it. It's so small. But a little bit of mucus is coming up. You're like, that's really disgusting. And then what do you do with the mucus? Yeah, you just swallow it. You don't even think about it. You're like, oh, now I'm thinking about it. Yeah, you're welcome. You're welcome for that. Transitional epithelium. Transitional is a special case. It stretches from a cuboid to flattened squamous. And it only is found in the bladder. And so it moves from this left image to this lower right image in the bladder, allowing for the accommodation of fluid as your bladder stretches. This is a histological preparation. There's an optional DVD. I was talking to some of your colleagues. If you want more histology, there's these types of histology on the DVD. You do not need it. You don't need it for lab, but if you're interested in understanding it to a greater extent, that is available in the bookstore. But like, so for example, I'll pull the picture here to show you, so you don't have to study extra. You'll just see the pictures here. But this is an example, and you'll see this in lab. Transitional epithelium is found in the bladder where it actually can go from a box shape to a flattened structure as it stretches. It's really kind of cool. So again, one of the best examples that I can think of structure leading to functionality. Because can you imagine if your bladder didn't stretch? I mean, 25% of you would be rotating out to go to the bathroom. Fortunately, it's right there. And then the next 25% would be going in an hour and 15-minute lecture. But, even after your monster drink, it'll accommodate a little bit of stretch. You can hold it until the end of lecture. Okay. Functions for simple epithelium. Well, simple epithelium, we've already kind of introduced absorption, secretion, diffusion. This can be simple squamous, simple cuboidal, or simple columnar. And you find that in the digestive tract, the kidneys, for simple cuboidal or columnar. Simple squamous, what was an example of simple squamous that we used? What's that? Trachea was pseudostratified columnar epithelium. Skin, remember the green picture down here? This is a blood vessel. Has a lumen, that's the white space. So you're actually looking end on in cross section down the blood vessel. So blood vessels or skin are great simple squamous. This is the ciliated trachea over here. That's pseudostratified. That's this far right one. And the simple cuboidal, these circular structures are renal tubules in the kidney, and they're these boxes. They're cuboidal in a circle. I think it looks like a pineapple slice, but that's just me, okay? Stratified epithelium. Well, stratified versus simple provides a lot of protection. Simple is absorption, secretion, Simple is absorption, secretion, and diffusion because it's one layer. It's easy to move something across it. Stratified is for protection. It's like for abrasion. So you're going to find it skin, mouth, esophagus, anus, vagina. Stratified, basal side is the regenerative side, and the apical side is what sloughs off and is replaced. Pretty straightforward, right? This stuff isn't that complicated if you spend the time with it. So the two pictures on the far left and in the middle are both pseudostratified columnar epithelium. These two. What is this one? It's stratified. Which one is it? Far right. What's that? It's stratified squamous. That's right. Where would you find it? Where's an example? This particular prep is from a specific location in the body. What's the most common stratified squamous example I've been using today? Skin. Yep. Questions? How are we doing? Making sense? Okay, we're going to be pivoting a little bit to connective tissue. What is the purpose of connective tissue? If I was to ask you what's the function of epithelium, I feel like you have an arsenal of things that you could say, well, it kind of depends. Keller, it kind of depends. Like, is it simple or is it stratified? And stratified is probably more for protection. It's simple, it could be secretion and absorption, right? But connective tissue is a little bit more straightforward. It's not quite as complicated as epithelium. I think epithelium is far more interesting, but that's just my personal bias. But connective tissue is important. What does it do? What did I tell you before about how creative scientists and engineers really are when they name things? What was that? It connects things. Oh, man, brilliant. Absolutely brilliant. It does exactly that. It actually connects and binds things together. It glues tissues into the organs themselves. It binds bones to bones. It binds muscles to bones. What's a name for a structure that connects a muscle to a bone? A tendon. What's the name of a structure that connects bones to bones? A ligament. Those are types of connective tissue. Make sense? Like you need your muscle to connect to your bone, and sometimes you need your bone connect to your other bone. Now, we are going to define these things by their cell and matrix types as we investigate connective tissue. So what the heck is this picture? Anybody know what this picture is? It's not a tattoo, but it's close. It's in a similar category. So it's actually, what's that? It's essentially a forced scar. It could be a keloid in certain uh, individuals that are prone for keloid formation. In ethnic skin is definitely more prone to keloid formation. So this is a process or a tradition pronounced as citricization, where it's inducing wounds in skin to create a pattern. As it heals and scars, it can form elaborate patterns. And tribes, historically, indigenous tribes in Africa where this was invented, would use that to identify one tribe versus another. Okay? But it's a really interesting picture, and I think with the amount of, body artwork that's happening today, we're seeing some of this manifest itself more mainstream society today. But it's basically connective tissue as it's being laid down post embryonic development in a wound healing process, creating artwork or a tattoo-like architecture. So citrice is the French term, which is a Latin derivative for the word scar. Citrice. So citricization is what you're seeing here in this particular picture. Okay, now that's just to get you kind of interested in, some of you have seen that before, right? You've seen individuals with that kind of elaborate pattern, or maybe not quite the whole back, but okay. Connective tissue, what does it do? Well, it supports, it protects, it cushions, it insulates. So you can see this picture here between bone to bone. You have articulating cartilage that forms this nice, soft cushion. And as we age, that articular cartilage wears out. And now you have knee problems or joint problems, right? Your dad or your mom, your uncle, grandparents complain of, their knees hurting. Most of you are probably too young for this, but it'll happen, I promise, especially if you're active. Some of you are shaking your head like you've already had this issue, okay? Yeah. So supporting things like organ capsules and bones, like around an organ. It's like an Amazon package comes to you, right? And it's something that's fragile. You open it up and you're kind of hoping that you're pulling out like either, popcorn stuffing or the bubble wrap or, crumpled up brown paper, right? And you're happy, you're really satisfied when you see a lot of it. You're like, oh, they did a nice job. Same thing with valuable organs. There is connective tissue that surrounds those organs. A lot of that is fat or adipose tissue that surrounds organs. But fat, adipose tissue, is a type of connective tissue. We'll get there. Cushioning and insulating. Loose connective tissue, like within the skin. You want it to be loose because you need it to stretch, right? If you're going to have an abrasion here, well, if it doesn't give, it's just going to tear. But if it gives a little bit and it stretches, it's actually more resilient. And you lose that elasticity as you age. That's why older patients bruise easily. They bump themselves and they tear their skin. And if you're in that field of treating our geriatric population or you have aging grandparents, you're like, it's amazing how thin their skin is. You know, for you and I'll put myself in your category, right? We're not there yet, right? Like it's still thick enough where it's resilient, and it probably will heal pretty quickly. You cut yourself, a couple of days, you're like, oh, it's already closed up. But that process of healing actually is compromised as we age. Adipose tissue, coming from adipocytes, fat cells. Adipo for fat, cytes for cells, that insulates and cushions the organs like we just talked about. Okay, so on this slide, we've got a couple of different images of connective tissue. Another feature of it, storing and transporting nutrients, like adipocytes store fatty acids. Bone marrow, somebody said bone marrow earlier. I don't remember what the question was, but bone marrow is a type of connective tissue. It stores blood cell precursors. It stores the stem cell population that gives rise to your red blood cells and your white blood cells. This is why if you have a patient who's struggling with a leukemia or a blood disease, a blood cancer, and it's in the marrow of the bone, then they won't make white blood cells, which lead to your immune system. Okay? If they can't make the red blood cells, they're going to be iron deficient or under-oxygenated. Bone itself stores calcium. Now, calcium is a really important compound for our body. It's important not just for the makeup of bones. You know that from all the milk commercials, maybe when you were a kid. Drink more milk, right? But we will talk about bone health in the skeletal system. You'll talk about it in lab as well, because the bones themselves are a living tissue. And they store calcium and they also release calcium, because you need calcium in your bloodstream for other physiologic processes. Like, so for example, you need calcium available to contract a muscle. You're like, really? Yeah. So the calcium that's stored in bones can be readily available in the circulation if your calcium levels are low and your muscle contraction is compromised. You're like, well, wouldn't you just like not do the exercise? Well, think about the other muscles like respiratory muscles, right? Think about the cardiac muscle. Like, even if you're just sitting there gaming, doing nothing, muscles are still contracting, keeping you alive. So calcium homeostasis is really important. It's really important. We'll come back to it in the skeletal system, and we'll come back to it when we talk about muscle physiology. But just remember for now that the bones store copious amounts of calcium. Now the blood, this is an interesting one. Blood is a connective tissue. Blood in you and I right now is in a liquid phase. But what happens when you have a cut? You want it to quickly go from a liquid phase to a solid phase, right? So it is a liquid connective tissue. And it has the ability to form a network spontaneously and link up and connect things and form a clot. So it's a really sophisticated connective tissue that it's biphasic. So in these pictures, upper right, what is this tissue? Adipose, fat tissue. Kind of a classic picture of fat. A large open vacuole in the middle. The cell nucleus is on the edge, kind of shoved over to the side. And they're very round in structure, storing lots of fatty acids. No water. Fat is one of the most efficient ways to store energy because it's very light. You're like, what? Fat is very light because there's no water in it. It's hydrophobic, and water is extremely heavy. So if you look at individuals that are elite athletes that have high muscle content and very low fat, they actually weigh a lot. They're pretty heavy on a scale, a lot of mass. Fat, your body chooses to store it because you don't need to use water to store it. And you can make the calorie energy available pretty quickly. Okay, lower left, what is that tissue? Just identification. Bone. It's bone. Middle, Red blood cells, blood. Far right, lower right, bone marrow. Nicely done, bone marrow. The A is pointing to adipose tissue in the bone, and the T arrow is pointing to the trabeculae, or the solid bone structures. So the solid bone structures in the marrow are less prevalent than the rest, which is mostly fat. Bone marrow is mostly made up of fat. Okay, characteristics of connective tissue. Do you remember how I told you that it's cell poor and it's matrix rich? So immature cells are called blasts, and mature cells are called sites. So you're going to see a lot of terminology like an osteocyte or an osteoblast. That's a bone cell telling you if it's a mature or immature. The matrix component, this is outside the cell, so it's what we refer to as extracellular matrix. Outside, extracellular. And this matrix, this picture here, is areolar connective tissue. And you can see I've labeled the pink thick bundle, or the pink thick fiber, is a collagen fiber. And the dark black thin line is an elastic fiber. And so this is what connective tissue is made up of, is collagen and elastin primarily, with some other things that we'll learn about. Connective tissue is extremely well vascularized, different than epithelial tissue. Do you remember that? Epithelial tissue is not well vascularized. Oxygen gets there through diffusion. With connective tissue, it's typically extremely well vascularized, except for two examples that will be easy for you to remember. Tendons and ligaments. The reason it'll be easy to remember is when you have an injury to a tissue, if it's well vascularized, it heals very quickly. That's why skin wounds heal fast in most healthy patients. Bone fractures, bone is extremely well vascularized, heals really fast. How about a ligament or a tendon injury? To my athletes in the room, how well does that heal? How fast does that heal? Yeah, everyone's shaking their head, right? I mean, this is almost a scientific experiment. I could do some counting, I could run statistics, and you guys could agree with this statement. Just don't believe me. You could actually see the data for yourselves. Tendons and ligaments are poorly vascularized, and that's why they don't heal well. Some literature refers to them as avascular. I don't agree. There's a small amount of red zone in these tissues that has some amount of perfusion, but for the most part, in comparison to like muscle or bone, they're very, very poorly vascularized. The matrix itself has what we refer to as a gel component. This gel component provides a form. It provides the opportunity for water to be in the tissue itself. Remember in the early lectures we were talking about the composition of the body being mostly water, right? How much water is in the body on average? Give me a range. Yeah, 75% plus or minus. If you're dehydrated, obviously it's less, right? If you've been well hydrated, it might be 75, 77. But where does that water sit? Well, it sits throughout the entire body in the extracellular matrix. So the gel component is made up of what we refer to as polysaccharides. Polysaccharides are attached to a protein. So that protein has a saccharide or a sugar molecule that's attached to it. So we can refer to these as a proteoglycan, a protein sugar, proteoglycan. You following me? The proteoglycan has a net negative charge. It has a net negative charge, so it attracts a positive molecule. What's an example of a positive molecule that's high concentration outside of the cell in the extracellular matrix? What's that?
Water.
No, not water. What's an ion that would be it's a good guess, but that's what we're trying to get. We're trying to attract water. So we can't use water to attract water. It is sodium. Do you know why sodium's high outside the cell? What's responsible for making sodium high outside the cell? Sodium potassium ATPase, that thing again. 3 sodiums out, 2 potassiums in. A lot of salt, a lot of sodium, shouldn't say salt, a lot of sodium outside the cell. That's attracted to a net negative charge of the proteoglycan, right? Because it's sodium's +1. The proteoglycan is negatively charged, it attracts the sodium. What does water like to do when there's high sodium? Water likes to do what? It likes to follow the sodium. So now the water follows the high sodium concentration outside the cell that's bound to the proteoglycan. Proteoglycan has sodium attached to it because it's negatively charged. Sodium is positively charged. That moves water into the extracellular matrix. That's why your tissues are hydrated, because of these guys, the gel component. Make sense? So the chemistry actually, if you understand the chemistry of like sodium is +1, a proteoglycan is a sugar protein, it's net negative charge, it attracts the sodium. Now the principle of osmosis, water follows solutes, water follows where the sodium is outside the cell. That creates a moist environment surrounding the cell. We just put a bunch of things together over the last couple of weeks right there with this particular slide. You follow that? So it's sponge-like. It's literally like a sponge. It's on your kitchen counter. It's not all dried out. It's like after you got done with the dishes, you wring it out, and you set it there. It's still moist, right? You could probably wring it again and get a couple more drops of water. So the gel component of the extracellular matrix houses most of that water that we're quoting that makes up the body. This functions to resist compression, right? Like a wet sponge. It's kind of squishy. Your tissues are squishy, to use a technical term. It creates pores and spaces because it's a swollen tissue. Okay. Fibrous component of the matrix is made up of collagen. And this collagen, also elastin, we'll cover elastin on the next slide, but the collagen is the most abundant protein in extracellular matrix. The collagen on this areolar connective tissue is the pink wide fibers. But collagen, there's 28, I just looked it up today. Last time I lectured, I think there was only 26 discovered. But now there's 28 different variants of collagen in the body. And they're fashioned like you would take a piece of rope, 3 pieces of rope, and braid it. And so you have a fat rope with three strands that are braided. How strong is that? Extremely strong. Stronger than 1, stronger than 2, stronger than 3 if you just put them together. If you braid them, right, it's really, really strong. So that's the microarchitecture of collagen, is braided 3 collagen fibrils. So a lot of strength, an absolutely tremendous amount of strength. Now, the other fibrous component in the matrix is elastic fibers. And the elastic fibers are the green one. This squiggly green this arrow that's pointing to the squiggly black line, that's the elastin fiber, which is thread-like and recoils and stretches like a rubber band would do. So you have 2 different components. You have strength from the collagen, the 3 braided strands, and you have elastomeric recoil that allows your skin, when you pull it, to let it go and it snaps back. One test that we do clinically for patients to see if they're actually dehydrated is you actually grab the skin, pinch it, and let go. And if it kind of stays molded, that means there's not enough gel component in the extracellular matrix, not enough fluid, not enough water. Okay, so just for sake of completeness, the yellow arrow is pointing to the reticular fiber. So you've got the red arrow pointing to collagen up top. You've got the green arrow pointing to elastin and the yellow arrow pointing to the reticular fiber, which is about half the thickness of the elastin fiber. So different combinations of matrix in the body. Different combinations of matrix in the body. We can have more elastin than collagen. The first line, they would be very elasticity would be very high in the tissue. Like bladder is that way. Lots of stretch in the bladder. The aorta has a lot of elastin. Bones have about 6 to 9% elastin. That's why they don't just snap all the time. Okay, you have to really push them. But they do flex. Bones do. They're not like a metal rod. Collagen greater than elastin, more tensile strength. If you have a larger gel component over the fibrous, like more of the proteoglycans over elastin or collagen, then you get more cushioning, more support. Pretty straightforward. So I do want you, for exam 1, to be able to compare and contrast the characteristics of epithelial and connective tissue. I do want you to be comfortable with the histology. You'll be reinforcing that in lab this week, which means you're kind of studying for 2 classes at the same time, which is really nice. Okay?